Okay, so I guess this is a very different topic from the ones that were going on for the last 1 hour. I initially thought that I would not get time to thank the organizers, but now I can. It's the last talk of the day, so I hope you are all alive and awake. So if you are all alive and awake, then I'll want to first thank the organizers for giving me this opportunity to present the work that I have been doing with my collaborator Abhishek Chaudhuri, who is here and going to present his own work tomorrow from IOP, and Nisha Gupta, who is a PhD student of mine, who has given the poster. If some of you have seen it, then you would already know what I'm going to talk about. In case not, then you should go and visit the poster. Okay, so the work is on a semi-flexible filament driven by molecular motors. The motivation behind that talk is basically the cellular cytoskeleton, which we know is a very complex and dynamic network of semi-flexible filaments of varying bending rigidity. So we have actin filaments, microtubules, intermediate filaments. They differ in their persistence lengths, and in fact, this is the persistence length for this would be about in the order of microns. This is millimeters, so you can imagine that the difference in scales. So here we see a fluorescently labeled picture of a cell where the fluorescent label is for the different filaments. These are the microtubules, and the actin filaments are labeled in red. As you can imagine, they are more close to the cell cortex. This, which is a movie which I couldn't play because of some technological problem, but what you would see is that these are organelles which are moving on cellular tracks on these microtubule tracks, and the microtubules themselves are also fluctuating. So it's not like you expect microtubules inside the cell to be extremely rigid, but in fact you would see that they are also have some floppiness. Okay. So obviously the cellular cytoskeleton is a very important part of the cell. It gives its cell its stability, structure, the shape of the cell, and performs a whole host of functions. Now the question, of course, is how much do we understand these things, these cytoskeletal components? And in fact, it turns out that a large number of information about the cytoskeletal components, which comprises of these filaments and these motor proteins, which are which are active. These are rendered active by hydrolysis of this chemical fuel, either GP or ATP. Now, all this information comes out from a lot of these information comes from in vitro experiments. In fact, this is one of the very interesting in vitro setups. This is called a gliding assay, where basically these filaments are grown outside the cell, and they are capped so that they are you do not see a change of the length. Otherwise, they could inside the cell they do polymerize and depolymerize. But in this case, they are capped, and they are placed on a bed of motor proteins. So they are moving on a bed of motor proteins. These motor proteins inside the cell would take the cargo carry the cargo and move ahead in the process on the tracks. However, in this case, what is done is basically it is inverted, and you have this the head is now irreversibly attached to this glass slides, and the legs are dangling around. So you have these filaments moving on these motor proteins, and at sufficiently high densities you see some fascinating structures. In fact, you see this self-organization of these filaments, and they move as as swirls. In fact, if you look at these are two different set of filaments. This is actin filament, this is microtubules, and depending upon their which filament and the corresponding motor proteins that you have, you can see these swirls, or you can in fact see for the case of let me just play this movie. Doesn't. Okay. Anyway, so you would have seen that this actually forms very fantastic vortices, and these are like each of these vortex could be of the order of 40 microns in diameter. That's like really significant. I mean, this and the filaments move around in this inside these vortices. Okay, so so the next question, of course, is what we wanted to do was to see how these behaviors can be understood, and so we set up a computational model where we basically modeled these filaments as a worm-like chain, and these motor proteins which are stuck on the substrate. These were modeled as elastic springs. So this is a work that I had presented before in one of this ISPS PCM meetings, in fact in 2016. And what we did there was to put this motor proteins on a line, and then ask this question that what happens when you start pulling this filament out of the substrate. So this filament, obviously what this motor proteins does is this attach and detach to these filaments. This and if they are attached to this filament, then they will move with a certain velocity on the filament, and that will, as a result of that, there will be a load force which will act, and therefore it can detach. This will be attachment and detachment rates. And now the question is, this is of course an active system, and in such a scenario, what happens to this adsorption-desorption transition? And in fact, we showed that the adsorption-desorption transition is significantly affected by the active velocity of the motor proteins, as well as the attachment-detachment kinetics, which is signified by this this ratio, which is this dimensionless ratio. Okay, in fact, we could also do an analytic calculation where which matches quite well with the experimental with the simulation results. So this was done, and then subsequently then they asked the question that what is that's a scenario which I showed you in the first couple of movies. The experimental scenario of having this on a 2D substrate. Now you have filaments now you have motor proteins which are decorated on a 2-dimensional substrate, and you ask what happens when you have a filament which is a worm-like chain placed on this substrate. Okay, so here what you see, what I'm showing you are two different morphologies of this filament for different conditions, and you can see that it can switch between what are open states and coiled states. So they can also coil up. So that's significant. Okay, having said that, so let's ask what are the two modes of activity. The two modes of activity are this detachment rate, which is of course dependent upon the local load, and the active velocity, which of course again is dependent upon the local load. Okay. Now, in order to understand this effect of local stress on the conformational properties of the polymer, what we do is we play around with these rates. Okay. So in one case, what we do is to say that there is no dependence on the local load, which means that the off rate is basically a bare, it's a constant detachment rate. Okay. Otherwise, it would have been something which would depend upon the load, because obviously if the motor protein gets stretched, then you would expect that the off rate should increase with the increasing load force. So if we do that, then what do we see? Okay. So here it shows these are end-to-end distributions of and the probable distributions of the end-to-end vector, and then what we find is that it's actually significantly affected. So let me just spend 2 minutes on this slide. Here you see that what we are doing is we have kept the attachment-detachment rate to be the detachment rate to be load independent, which means that it's a constant detachment rate. Okay. Now imagine that we have no velocity of this motor proteins, which means that the velocity of the motor proteins is 0, which means that the motor proteins do not walk. What it does is basically they do attachment and detachment with a constant rate. If we do so, then we find that this is actually the ratio of the end-to-end probable distribution to the equilibrium probable distribution. This is the equilibrium probable distribution of the polymer, of the semiflexible polymer, and you find that there is a dip near near r tilde equal to 0, which basically signifies that what has happened is that the polymer is now become more stiff as compared to the equilibrium polymer. Okay. Now the moment we switch on this active velocity, just the moment we go from 0 to V0, so you put a constant active velocity, not a load dependent one, then you find that immediately it shows a very interesting behavior, that it just coils up. Okay. This coiling up shows that it's basically softens. The polymer effectively softens and it coils up, and shows this kind of a behavior. So this probable distribution shows this kind of behavior. Okay. Now if you start increasing the, if you put in the load, if you put in a velocity which is dependent on the load, again you see a peak near that, which basically shows that it goes between open and coiled states. Okay. Now you can obviously play around with this system. You can ask what happens when you have both detachment and active velocity stress dependent, which means that both depend upon the load, which is kind of presumably the more experimentally relevant scenario. And in this case, of course, it goes very much like this. The behavior is very close to that of the equilibrium polymer for very small activity. This Peclet is basically the ratio of the dimensionless ratio of the advection to the diffusion. And now if you go to very high Pecklets, what you observe is that there is again this coiled up states, and so this emergence of coiled up states happens again at large values of activity. Okay, so very little time left. So again you can look at tangent correlations, and you see that at very large Peclet you can see these oscillations in the tangent correlations, which is kind of expected because it goes between open and closed states, between this open and coiled states. One more interesting property which I just need to which I want to specify for this particular case. It's a very interesting property that we observe, and it's a dynamical property. Here what we are looking at is the mean square displacement of the center of mass. Okay. Now what we know that in case of either for persistent random walkers or directed random walkers, you know that there is only one crossover, either from a diffusive to ballistic or from ballistic to diffusive. What you observe here in this particular case is that a series of such crossovers. Okay. The mean square displacement shows crossovers from ballistic, diffusive, and so on. Okay. Now the question is, why does it happen? What happens is, in this case what we find is that there are two time scales, additional time scales which are there. One is the persistence of the direction of the center of mass velocity, and the other is the correlated fluctuations of the center of mass speed. So there is a speed as well as the correlation as well as the orientation of the center of mass velocity, and that you can see from this correlation, and you can see that there is actually the decay of this correlation gives you that correlation time, and you have multiple decays, exponential decays that you can observe. Okay, so that gives rise to this series of crossovers, which otherwise would not have been, you wouldn't see in normal active polymer models, which model the polymer as a constant active force which is acting on each of these beads. So there we conclude that the distance from equilibrium is reflected in the end-to-end distribution, modified bending stiffness, and a transition to spiral morphology of the polymer. The local stress dependence of activity results in correlated fluctuations in the speed and direction of the center of mass, which leads to a series of ballistic diffusive crossover in its dynamics. So hopefully in the next ISPCM we will be able to show more on collective dynamics of these polymers. Thank you.
Questions? I have one question. So finally, are you saying there is some, I mean, effect, like I didn't understand, it's in the end-to-end like the tangent correlations.
Yeah.
You see the oscillations.
Yes, that's true. So these show, I mean, if you are at very low Peclet, then you don't see, that's an exponentially it goes down. Whereas when you go to high Peclet, the high Peclet, remember, is showing you, it goes between a coiled up state and an open state. So yeah, this fluctuation, this keeps on happening, and that periodicity is picked up. I mean, that is picked up in this tangent correlation. Yeah. So I mean, well, if you look at the long-term dynamics of this, you will see that this is like the end-to-end distance, and if you look at that, then you will see that this. It goes between this open and coiled up states. It keeps happening. Yeah. At least, okay. So you are saying that whether this period has, whether it's the same period or not. Yeah. Well, could be. I mean. Yeah. What is the frequency? So that's something, of course. Yeah. Coil, yeah, absolutely. So it goes on, it just keeps on doing that. Yeah. Yeah. Okay. Yeah, it's a tangent correlation in S. Is this one? Yeah. Sorry, I should have said that. It's tangent correlations is in the. But then it doesn't correspond to open close. No, this is a coil which is picking, which it is picking up. Yeah. Sorry, there was another one which also shows the temporal one, but this is not the one. Yeah. So this is the S. Okay. Yeah.
So if there is no other question, let's thank all the speakers and the audience.